cardiovascular system in overall body homeostasis. Identify the major body fluid compartments and state the approximate volume of each. Diagram the blood flow pathway between the heart and other major body organs. How does the arrangement of heart and vessels enable unidirectional flow of well-oxygenated blood to the body? What is the advantage of the reciprocal relation between blood flow velocity and vascular cross-section area? How do the differing compositions, smooth muscles, fibrous and elastic tissue of blood vessels contribute to their respective functions? Circulatory system transports and distributes essential substances to tissues and removes metabolic byproducts. The system also participates in homeostatic mechanisms such as regulation of body temperature, maintenance of fluid balance, and adjustment of O2 and nutrient supply in various physiological states. The cardiovascular system, which accomplishes these tasks, is composed of a pump, the heart, a series of distributing and collecting tubes, blood vessels, and an extensive system of thin vessels, capillaries, that enable rapid exchange between the tissues and vascular channels. Blood vessels throughout the body are filled with the heterogeneous fluid, blood, that is essential for the transport processes performed by the heart and blood vessels. Three compartments of watery fluids, known collectively as the total body water, account for about 60% of body weight. This water is distributed among the intracellular, intestinal, and plasma compartments. Know that about 2/3 of our body water is contained within cells and communicates with the intestinal fluid across the plasma membranes of cells. Of the fluids that is outside cells, that is extracellular fluid, only a small amount. The plasma volume circulates within the cardiovascular system. Blood is composed of plasma and roughly an equal volume of four elements, primarily red cells. The circulating plasma fluid communicates with the intestinal fluid across the walls of small capillary vessels within organs. The intestinal fluid is the immediate environment of individual cells. The cells must draw their nutrients from and release their products into the interstitial fluid. The interstitial fluid cannot, however, be considered a large reservoir for nutrients or large sink for metabolic products, since its volume is less than half that of the cells that it serves. The well-being of individual cells therefore depends heavily on the homeostatic mechanism that regulate the composition of the interstitial fluid. This task is accomplished by continuously exposing the interstitial fluid to fresh circulating plasma fluid. As blood passes through capillaries, solutes exchange between plasma and interstitial fluid by the process of diffusion. The net result of transcapillary diffusion is always that interstitial fluid tends to take on the composition of the incoming blood. If, for example, potassium ion concentration in the interstitial fluid of particular skeletal muscles were higher than that in the plasma entering the muscle, potassium would diffuse into the blood as it passes through the muscle's capillaries. Since this removes potassium from interstitial fluid, interstitial potassium ion concentration would decrease. It would stop decreasing when net movement of potassium into capillaries no longer occurred, when interstitial concentration reached that of incoming plasma. Three conditions are essential for this circulatory mechanism to effectively control the composition of interstitial fluid. There must be adequate blood flow through the tissue capillaries. The chemical composition of the incoming or arterial blood must be controlled to be that which is optimal in the interstitial fluid. And third, diffusion distance must be short. Transport occurs over extremely small distance because almost no cells in the body is located further than about 1 micrometer from a capillary. Over such microscopic distance, diffusion is a very rapid process. However, it's a very poor mechanism by which to move substances from the capillaries of one organ, such as the lungs, to the capillaries of another organ that may be 1 meter or more distant. Consequently, substances are transported between organs by the process of convection, by which the substances move along with blood flow simply because they are dissolved or otherwise contained within blood. It is common practice to view the cardiovascular system as the pulmonary circulation, composed of the right heart pump and the lungs, and the systematic circulation, in which the left heart pump supplies blood to the systemic organs, all structures, except the gas exchange portion of the lungs. The pulmonary and systemic circulation are arranged in series, that is, one after the other. Consequently, the right and left hearts must each pump the same volume of blood each minute. This amount is called the cardiac output. A cardiac output of 5 to 6 liters per minute is normal for a resting individual. Thus, for example, the cardiovascular response to whole body exercise can involve increased blood flow through some organs, but decreased or unchanged blood flow through others. Many of the organs in the body help perform the task of continuously reconditioning the blood circulating in the cardiovascular system. Organs that communicate with the external environment, such as the lungs, play key roles. Like the lungs, many of systemic organs will also serve to recondition the composition of blood, while the flow circuitry precludes their doing so each time the blood completes one circuit. The kidneys, for example, adjust the electrolyte composition of the blood passing through them. Because the blood conditioned by the kidneys mixed freely with all the circulating blood, and because electrolytes and water freely pass through most capillary walls. The kidneys control the electrolyte balance of the entire internal environment. To achieve this, is it necessary that given until blood pass often through the kidneys. In fact, the kidneys normally receive about 1/5 of the cardiac output. This greatly exceeds the amount of flow that is necessary to supply the nutrients needs of the renal tissue. This situation is common to organs that have a blood conditioning function. Blood conditioning organs can also withdraw, at least temporarily, severe reduction of blood flow. Skin, for example, can easily tolerate a large reduction in blood flow when it is necessary to conserve body heat. Most of the large abdominal organs will also fall into this category. The reason is simply that because of their blood conditioning functions, their normal blood flow is far in excess of that necessary to maintain their basal metabolic needs. The brain, heart muscle, and skeletal muscles typify organs in which blood flows primarily to supply the metabolic needs of the tissue. They do not recondition blood for the benefit of any other organ. Flow to brain and heart muscle is normally only slightly greater than that required for the metabolism, and they do not tolerate blood flow interruption well. Unconsciousness can occur within a few seconds after stoppage of cerebral flow, and permanent brain damage can occur as little as 4 minutes without flow. Similarly, the heart muscle, myocardium, normally consumed about 75% of the oxygen supplied to it, and the heart's pumping ability begins to deteriorate within bits of a coronary flow interruption. The task of providing adequate blood flow to the brain and the heart muscle receives a high priority in the overall operation of the cardiovascular system. The heart consists of 2 pumps in series. One pump propels blood through the lungs for exchange of oxygen and carboxygen, the pulmonary circulation, and the other pump propels blood to all other tissues of the body, which name the systemic circulation. Flow of blood through the heart is in one directional or unidirectional. Unidirectional flow through the heart is achieved by the appropriate arrangement of valves. Also, cardiac output is intermittent, continuous flow to body tissues periphery occur by distension of the aorta and its branches during ventricular contraction systole, and by elastic recoil of the walls of the large arteries, which forward propulsion of the blood during ventricular relaxation or diastole. Cardiac muscle functions as a syncytium, that is, a stimulus applied to any part of the cardiac muscle results in contraction of the entire muscle. Gap junctions with high conductance are present in interscalated disc between adjacent cells and facilitate conduction of the cardiac impulse from one cell to the next. Cardiac muscle must contract repetitively for a lifetime, and hence it requires a continuous supply of oxygen. To provide adequate oxygen and substrate for its metabolic machinery, the myocardium is also endowed with a rich capillary supply, approximately 1 capillary per fiber. Thus, diffusion distance are short, and oxygen, carboxygen, substrates, and waste material can move rapidly between the myocardial cell and capillary. The transverse tubular system within myocardial cells participates in this exchange of substances between capillary blood and myocardial cells. The T-tubular system is absent or poorly developed in the atrial cells of many mammals. The atria are thin-walled, low-pressure chambers that function more as large reservoir conduits of blood for their respective ventricles than as important pumps for the forward propulsion of blood. The ventricles comprise a continuum of muscle fibers originating from the fibrous skeleton in the base of the heart, chiefly around the aortic orifice. These fibers sweep toward the cardiac apex at the epicardial surface. They pass toward the endocardium and gradually undergo an 180-degree change in direction to lie parallel to the epicardial fibers and to form the endocardium and papillary muscles. At the apex of the heart, the fibers twist and turn inward to form papillary muscles. At the base of the heart and around the valve orifices, these myocardial fibers form a thick, powerful muscle mass that not only decrease the ventricular circumference to implement the ejection of blood, but also narrow the atrioventricular valve orifices, which aids in closure of the valve. Ventricular ejection is also accomplished by a decrease in the longitudinal axis as the heart begins to narrow toward the base. The early contraction of the apical part of the ventricles, coupled with approximation of the ventricular valves, propels the blood toward the ventricular outflow tracts. The right ventricle, which develops a mean pressure that is approximately 1/7 that developed by the left ventricle, is considerably thinner than the left ventricle. The cardiac valve leaflets consist of thin flaps of flexible, tough, endothelium-covered fibrous tissue that are firmly attached at the base of the fibrous valve rings. Movement of the valve leaflets is essentially passive, and the orientation of the cardiac valves is responsible for the unidirectional flow of blood through the heart. There are two types of valves in the heart: atrioventricular and semilunar. Atrioventricular valves. The tricuspid valve, located between the right atrium and the right ventricle, is made up of three cusps, whereas the mitral valve, which lies between the left atrium and the left ventricle, has two cusps. The total area of cusps of each atrioventricular valve is approximately twice that of the respective atrioventricular orifices, and so considerable overlap of the leaflets occurs when the valves are in the closed position. Attached to the three edges of these valves, a fine, strong ligament called the tendinea cordis that arise from powerful papillary muscles of the respective ventricles. Those ligaments prevent the valves from becoming everted during ventricular systole. In a normal heart, the valve leaflets remain relatively closer together during ventricular fillings. The partial approximation of the valve surface during diastole is caused by eddy currents that prevail behind the leaflets and by tension that is exerted by the tendinea cordis and papillary muscle. Semilunar valves. The pulmonic and aortic valves are located between the right ventricle and the pulmonary artery and between the left ventricle and the aorta. These valves consist of three cup-like cusps that are attached to the valve ring. At the end of the reduced ejection phase of the ventricle systole, blood flow briefly reverses toward the ventricles. This reversal of blood flow snaps the cups together and prevents regurgitation of blood into the ventricles. During ventricular systole, the cusps do not lie back against the walls of the pulmonary artery and aorta. Instead, they float in the bloodstream at a point approximately midway between the vessel walls and their closer position. Behind the semi-lunar valves are small outpocketing sinuses of valsalva of the pulmonary artery and aorta. And these sinuses, eddy currents develop, which tend to keep the valve cusp away from the vessel walls. Furthermore, the orifices of the right and left coronary arteries are behind the right and the left cusps, respectively, of the aortic valve. Were it not for the presence of the sinus of Valsalva and the eddy currents developed therein, the coronary ostia could be blocked by the valve cusps, and coronary blood flow would cease. The pericardium invests the entire heart and the cardiac portion of the great vessels, and it is reflected onto the cardiac surface at the epicardium. The sac normally contains a small amount of fluid, which provides lubrication for the continuous movement of the end-closer heart. The pericardium is not very distensible. It strongly resists a large, rapid increase in cardiac size and hence prevents sudden over-distension of the chambers of the heart. However, in congenital absence of the pericardium or after its surgical removal, cardiac function is not seriously affected. Nevertheless, with the pericardium intact, An increase in diastolic pressure in one ventricle increases the pressure and decreases the compliance of the aortic ventricle. In the normal intact circulation, the total volume of blood is constant. An increase in the volume of blood in one area must be accompanied by a decrease in another. However, the distribution of blood circulating to the different regions of the body is determined by the output of left ventricle and by contractile state of the resistant vessels, arterioles of these regions. The circulatory system is composed of conduits arranged in series and in parallel. This arrangement has important implications in terms of resistance, flow, and pressure in blood vessels. Blood entering the right ventricle via the right atrium is pumped through the pulmonary arterial system at mean pressure about 1/7 that is a systematic artery. The blood then passes through the lung capillaries, in which carboxygen in the blood is released and oxygen is taken up. The oxygen-rich blood returns via the pulmonary veins to the left atrium, where it's pumped from the ventricle to the periphery, which thus completes the cycle. Blood moves rapidly through the aorta and its arterial branches. As these branches approach the periphery, the branches narrow and their walls become thinner. They also change histologically. The aorta is predominantly elastic structure, but the peripheral arteries become more muscular until the arterioles, the muscular layer, predominates. In the large arteries, frictional resistance is relatively small, and pressure are only slightly less than those in the aorta. The small arteries, on the other hand, offer moderate resistance to blood flow. This resistance reached a maximal level in the arterioles, which are sometimes referred to as stopcocks of the vascular system. And the pressure drop in greater across the terminal segment of the small arteries and the arterioles. Adjustment in the degree of contraction of the circular muscle of these small vessels allows regulation of tissue blood flow and aids in the control of arterial blood pressure. In addition to the reduction in pressure along the arterioles, there is a change from pulsatile to steady blood flow. Pulsatile arterial blood flow, caused by the intermittent injection of blood from the heart, is damped at the capillary level by a combination of two factors: the distensibility of the large arteries and frictional resistance in the small arteries and arterioles. Many capillaries arise from each arteriole. The total cross-sectional area of the capillary bed is very large, despite the fact that cross-sectional area of each capillary is less than of each arteriole. As a result, blood flow velocity becomes quite slow in the capillaries, which is analogous to the decrease in velocity of flow in the wide regions of a river. Conditions in the capillaries are ideal for the exchange of diffusible substances between blood and tissue because capillaries consist of short tubes with walls that are only one cell thick and in which flow velocity is low. On its return to the heart from the capillaries, blood passes through venules and then through veins of increasing size. Pressure within these vessels progressively decreases until the blood reaches the right atrium. Near the heart, the number of veins decreases, the thickness and composition of the vein walls change, the total cross-sectional area of the venous channels diminishes, and the velocity of blood flow increases. The velocity of blood flow and the cross-sectional area at each level of the vasculature are essentially mirror image. Data from humans indicates that between the aorta and the capillaries, the total cross-sectional area increases above the 500-fold. The volume of blood in the systematic vascular system is greater in the veins and venules, 64%. Only 6% of total blood volume exists in the capillaries, and 14% of total blood volume is found in the aorta, arteries, and arterioles. In contrast, blood volume in the pulmonary vascular bed is about equal divided among the arterial, capillary, and venous vessels. The cross-sectional area of the vena cava is larger than that of the aorta. Therefore, the velocity of flow is slower in the vena cava than in the aorta. The cardiac muscle, like skeletal muscle, is striated due to the elderly structure of the actin and myosin filaments and the accessory proteins that stabilize the sarcomere. Like type 1 skeletal muscle, cardiac muscle appears to be red in color due to the high content of mitochondria and myoglobin and its blood supply. The heart uses large amount of ATP in between 60–100 times per minute during normal resting conditions. For the lifetime of the normal adult, an oxidative phosphorylation is the main source of that ATP, that the high myoglobin concentration and large mitochondrial content. There are estimates that the myocardial ATP pool turns over every 10 seconds. The heart is able to use any substrate provided to it in the blood, and uptake is dependent on the concentration of those substrates such as glucose, pyruvate, lactate, free fatty acids, and ketone bodies. Normally, fatty acid oxidation provides 60–90% of the ATP used by the adult heart. Like skeletal muscle, calcium is essential for contraction and is provided by excitation-contraction coupling. Also, cardiac muscle can contract spontaneously due to pacemaker activity in the sinoatrial node. The individual muscle cells, myocytes, normally contract only when an action potential is initiated by the conduction system present in the heart. and transmitted through the cells specialized to conduct action potentials rapidly. Cardiac muscle cells have gap junction through which cells communicate information about membrane potential. That is, if one cell depolarized, the adjacent cells will also depolarize due to communication through the gap junction. Thus, all cardiac myocytes in the atria contract together because of this unified contraction of the ventricles or the atria. The heart is said to be a functional syncytium. Since all ventricular muscle cells contract together, there is no type of special recruitment of the heart. The heart relies on other mechanisms of increased range of contraction. Cardiac muscle cells contract when calcium levels in the cells increase from approximately 0.1 micromole to 1 to 10 micromoles. The level of calcium present in the cytosol to initiate contraction has a profound effect on the strength of contractility. Excitation-contraction coupling in cardiac muscle varies somewhat from the process in skeletal muscle. The anatomy of the sarcomere-sarcoplasmic reticulum interaction is different. Dyads are founded rather than triads as in skeletal muscle. There is a less sarcoplasmic reticulum in cardiac muscles, so the calcium release process relies on entry of calcium into the cardiac cell through the voltage gate channels. These open when the membrane potential reaches approximately -40 millivolts. The calcium channels are also called slow or L-type calcium channels because they open more slowly than sodium channels and remain open longer, generally about 200 for 300 milliseconds. Therefore, the action potential in cardiac ventricular cells is much longer than the action potential in skeletal muscle, in which the calcium channels don't actually open. Calcium entry through the sarcoplasmic reticulum calcium channels is essential for contraction to occur. Absence of calcium in the extracellular fluid would prevent the heart from contracting. The process of excitation-contraction coupling is initiated by the pacemaker cells in the sinoatrial node that spontaneously generate action potential. Term slow action potential because they lack fast sodium channels, and depolarization is due to calcium entry through the slow calcium channels. Action potentials are transmitted through the action conduction fibers across the atrioventricular valves and finally to the conduction system in the ventricles. All ventricular muscle cells depolarized at the same time due to the rapid influx of sodium down as electrochemical gradient through sarcolemma fast sodium channels. When the membrane potential reaches -40 mV, the slow calcium channels open, allowing calcium to diffuse down its concentration gradient into the cytosol. Some of this calcium causes opening of ryanodine channels receptors on the sarcoplasmic reticulum, and calcium diffuses out of the sarcoplasmic reticulum down its concentration gradient. Some of the calcium from the sarcolemma binds to the troponin, as does all of the calcium released from the sarcoplasmic reticulum. Calcium binding to troponin results in a similar type of interaction of actin and myosin and cross-bridge cycling to that which occurs in skeletal muscle. Relaxation occurs when the calcium concentration in the cytosols is lowered by the calcium ATPase of the longitudinal part of the sarcoplasmic reticulum, pumping calcium back into the SR. When calcium levels decrease, the calcium diffuses from a troponin and the cell relaxes. Two other proteins are involved in the removing calcium from the cardiac cell. Since calcium enters the cell with each action potential, there must be a mechanism to remove calcium, or the cell calcium content would increase with each heartbeat. The sarcolemma contains a calcium ATPase that has a high affinity for calcium and can therefore pump calcium out of the cell, probably even during diastole. The other protein is the sodium-calcium exchanger. The exchanger operates on the basis of the sodium-ion gradient. The sodium-ion concentration is greater outside of the cell than in the cell. While the exchanger sodium ion enters the cell and calcium ion is removed from the cell. 3 sodium ions enter for every 1 calcium ion leaving the cell. Manipulation of sodium gradient can have significant effects on calcium extrusion from the cell and thus affect contraction. Since calcium levels change during each action potential, there is some evidence that increases in heart rate. More action potential per minute can increase calcium availability for contraction, thereby increasing the amount of tension that can be generated. This phenomenon is called the staircase phenomenon, or TREP. Physiologically, heart rate is altered by autonomic nerve system, modulation of sinoatrial node, firing rate, and the sympathetic nervous system, component of the autonomic nerve system, not only increases heart rate but also increases contractility. Therefore, the physiological role of TREP is difficult to assess independent of sympathetic nerve system modulation of heart rate and contractility. There are 2 additional variations in contraction that occur in cardiac muscle that don't occur in skeletal muscle. Phosphorylation of contractile proteins alters the strength of contraction in the heart. The heart is very responsible to the sympathetic nerve system, the fight or flight component of the autonomic nerve system. With activation of the sympathetic nerve system, beta-adrenergic receptors on the cardiac muscle cells are activated and intracellular signaling scheme results in production of IMP and activation of protein kinase A. Phosphorylation of protein follows. Several proteins involved in contraction are phosphorylated and their activity is altered and allow more calcium to enter the cell, and the strength of contraction is increased. A protein called phospholambin normally inhibits the sarcoplasmic reticulum calcium ATPase. When phospholambin is phosphorylated, it exerts less inhibition of the ATPase, so calcium uptake is enhanced. Phosphorylation of the calcium channels doesn't seem to occur in skeletal muscle, in which the maximum amount of calcium is released during each action potential and therefore cannot be increased. Remember that skeletal muscle has 2 sarcoplasmic reticular cisterns in conjunction with the T-tube, whereas cardiac muscles has only 1 cistern associated with the T-tubule. Skeletal muscle doesn't seem to have functional phospholamp bond, so the calcium ATP ACE activity is always operating in its maximum capacity. Increasing the amount of calcium entering the cytosol is an important mechanism for increasing strength of contraction. Removing calcium faster for relaxation, an important mechanism when the heart rate increasing with sympathetic nerve system stimulation, and there is less time during the contraction relaxation cycle. The strength of contraction in cardiac muscle can be altered by change in the initial or resting length of the muscle cells, preload, similar to the phenomenon in skeletal muscle. Cardiac muscle, unlike skeletal muscle, can have physiologic changes in the length of the muscle cells. For example, when the volume in the ventricle at the end of diastole is changed, the muscle cell length is changed in the same direction. Increased ventricular and diastolic volume results in increased ventricular muscle cell length prior to the onset of contraction. The heart normally operates at lower than maximal cell length of preload, whereas skeletal muscle usually works at maximal preloads. Also, passive tension properties of the heart differ from those of skeletal muscle. Skeletal muscle doesn't increase passive tension until the muscle cell length is close to the length that gives the maximum active tension. Cardiac muscle has passive tension even at low cell lengths. These differences are due to anatomical arrangements of the muscle cells with the non-contractile components in the muscle. Skeletal muscle is more distanceable than cardiac muscle. The principle for the length-tension relationship, as in skeletal muscle, is that the change in cells and sarcomere length alters the degree of overlap of the actin and myosin filaments, and therefore increases the potential for cross bridges to form. Changes in the resting length of the whole muscle are associated with proportional changes in the individual sarcomere lengths. Maximum tension development occurs in sarcomere lengths of 2.2 to 2.3 micrometer. At shorter sarcomere lengths, the opposite thin filaments may overlap with each other and interfere with interaction with myosin, and long sarcomere lengths overlap may be insufficient for optimal cross-bridge formation. More cross-bridge interaction leads to a stronger contraction. Two other forms may contribute to the length tension phenomenon in cardiac muscle. The second mechanism may result from a length-dependent change in calcium sensitivity of the myofilaments. For a similar cytosolic calcium concentration, a less stretched muscle develops less force than does a more stretched cardiac muscle preparation. This change in calcium sensitivity occurs immediately after change in length with no delay. The sensitivity of the contractile proteins, specifically troponin C, seems to increase a greater resting length. Finally, there is some evidence that the amount of calcium released from the sarcoplasmic reticulum is greater at longer resting length. How much these two factors contribute to the greater tension development is open to speculation since studies to demonstrate these two effects of lens on calcium dynamics are generally performed in isolated cells or organelles. In summary, the heart usually operates at lower than maximal preloads and therefore has reserve. Increasing muscle length can have a profound effect on strength of contraction that allows the heart to meet the demands of increased work such occurs during exercise. The effects of altered preload on heart function can also be observed with isotonic contractions that represent a better match to physiological contractions of the heart as a pump. The left ventricle must develop tension pressure to match the afterload or aortic pressure in order to open the aortic valve and then allow the shortening phase of the contraction to pump blood, stroke volume, into the aorta. Recall from the discussion of the skeletal muscle that there is an inverse relationship between afterload and velocity of shortening and therefore between afterload and shortening. Greater afterload results in less shortening. Using isotonic contractions, the effects of increased preload, that is more cross-bridges cycling on the force-velocity curve, can be analyzed. When shortening and velocity of shortening are measured as a function of afterload, higher afterloads results in less shortening. If the preload is increased from length 1 to length 2, and the same afterload contraction are studied from the higher preload, velocity of shortening is greater for each afterload. If more cross bridges can interact, there is a more myosin ATPase activity and therefore more energy from ATP hydrolysis available for contraction. The cross bridges develop the greater tension needed to match the greater afterload, and more energy is available for more shortening in a greater velocity of shortening to occur. As stated above, cardiac muscle can increase strength of contraction when the preload is increased, as demonstrated by the length-tension relationship. Another way that cardiac muscle can increase strength of contraction is by increase in cytosolic calcium, resulting in increased contractility. Contractility increases the velocity of cross-bridge cycling. Therefore, increasing contractility can alter shortening and the velocity of shortening. In comparing the force-velocity curve during enhanced contractility, one can notice that increasing contractility causes the entire force-velocity curve to shift to the right below the maximum isometric tension. This increased contractility should be compared to the curve generated at the same preload, the back curve. Generally, increases in contractility result in more rapid contractions such that indexes of speed of tension, development of maximum velocity of shortening, are used to indicate increases in contractility. The mechanism by which the contractions are stronger are, however, different. More optimum overlap of the actin and myosin filament mediates the preload effect, whereas more cytosolic calcium to induce more rapid cross-bridge cycling mediates the contractility effect. The effect of increasing contractility can also be demonstrated by looking at the lens-tangent relationship. The mechanism is increased in the calcium that result from the activation of beta-adrenergic receptors with the consequent production of cyclic AMP and activation of phosphorylation of the sarcolemma calcium channels by protein kinase A.